Hello students, welcome to Read MedPrep Academy channel. Today we are going to discuss in genetics principles of inheritance and variation part 1. In this we are going to talk about multiple alleles, ABO blood groups. What is genetics? Genetics is a branch of biology that deals with the study of heredity and variations. What does it do? It describes how the characteristics and features pass on from the parents to their offspring in each successive generation. What is a gene? The unit of heredity is called a gene. The gene is the inherited factor that determines the biological character or trait of an organism. In detail, genes are segments of DNA. Each gene contains the information about the certain character or trait. Genes are transcribed and translated by the cell to make proteins. Proteins create a visible phenotype. For example, one gene might code for an eye color. The gene is used by the cells to make proteins which create green pigment in our eyes. What is meant by variation? A variation is the degree by which the progeny differs from their parents. What is the genetic variation? Genetic variation refers to the diversity in gene frequencies. It refers to the differences between the individuals or the differences between the population. What is the cause of genetic variation? Mutation is the ultimate source of genetic variation. Other mechanisms that can cause genetic variation like sexual reproduction and genetic drift. Every person is unique in his skin color and the facial features. So the wide range of skin colors and facial features that are observed in human beings is a result of genetic variation. That's just a simple explanation of genetic variation. What is meant by allele? The genetic segregations in the Mendelian inheritance reveal that all genes have two alternate forms. One is a dominant and another is a recessive. The dominant and recessive alleles, example, tall versus dwarf. The tall is represented by capital T, which is dominant, and the dwarf is represented by small t, which is a recessive allele. What are the types of allele? The dominant allele is the normal allele or the wild allele, and the recessive allele is a mutant allele. All humans get two copies of every gene, one copy from each parent. The two copies don't have to be identical. So each variation of a gene is called an allele. Alleles create diversity. For example, if a gene contains an information of eye color, one allele might code for blue eyes and another allele might code for brown eyes. So what are alleles? For every gene, you have two of the same allele. Or you can have two different alleles. One is a recessive allele, the other one is a dominant allele. A recessive allele will only produce a phenotype if there is no dominant allele present. If a dominant allele is present, then the character will be dominant. In other words, you need two copies of the recessive allele in order to have the trait that it codes for. Recessive alleles are symbolized by lowercase letters. You see the table on the right side. The dominant allele represented by capital A and the recessive allele represented by a small a. The father and the mother having hemizygous where the capital A and the small a are present. The generation that is formed by combining two capital A's, one capital A, one small a, one capital A and another small a, and two small a. That means wherever capital A is present, it will produce only dominant character. For a recessive character to be expressed, two small a's are necessary or two recessive alleles are necessary. With this genotype, small a will have blue eyes. What are multiple alleles? A gene can mutate several times and produce several alternate forms. When two or more alleles of a gene that control a particular trait or character occupy the same locus on the homologous chromosome of an organism, they are called multiple alleles. What is meant by multiple allelism? Inheritance of these multiple alleles is called multiple allelism. Multiple alleles are useful in studying human blood groups, sex determination in man, insects and birds, sex-linked inherited traits, and many genetic disorders. Now let us discuss multiple allelism with respect to blood groups. Multiple allelism occurs in humans in the inheritance of different types of blood groups. The blood group inheritance in humans can be understood by learning about the antigens and antibodies, ABO antigens and the blood group antibodies. ABO blood types. Multiple allele inheritance occurs in ABO blood groups. Blood differs chemically from one person to another. Now, what are ABO antigens? When two different incompatible blood types are mixed, agglutination occurs. This is called clumping, where the erythrocytes join together. And the basis of these chemical differences is due to the presence of the antigens on the surface of the RBCs. These antigens are present in the membranes of the RBCs and the epithelial cells. So, mixing of the incompatible blood types can cause cross reaction. This agglutination can also cause hemolysis of RBCs, and possible patient death can occur because of transfusion reactions when incompatible blood is given. So we have to see that this cross reaction should be prevented. Now, coming to the discovery of ABO antigens, Karl Landsteiner discovered two kinds of antigens called antigen A and antigen B on the surface of the RBCs of the human blood. Types of blood groups. Based on the presence or absence of these antigens, three kinds of blood groups have been identified. Type A, Type B and Type O were recognized. Later, the fourth and the rarest group, called the AB group, which is considered to be the universal recipient, was discovered in 1902 by two of Landsteiner's students, Von de Castello and Adriano Sturli. So, some history regarding the discovery of ABO blood group system. Karl Landsteiner discovered the ABO blood group system in 1901. Adriano Sturli and Alfred Von de Castello, who were working under the Landsteiner and were students of Landsteiner, discovered type AB a year later in 1902. Landsteiner was awarded the 1930 Nobel Prize in Physiology or Medicine for his work and discovery of ABO blood group system. Inheritance of blood groups. Bernstein in 1925 discovered that the inheritance of different blood groups in human beings is determined by a number of multiple allelic series. Where are these alleles located? The three autosomal alleles located on the chromosome number 9 and are concerned with the determination of the blood group in any person. So, the alleles for blood groups are in the same place on the chromosome number 9, which is an autosome in the mother as well as in the father. However, the genes have a different code giving the different blood group. For example, you can see that the mother on the left side having A blood group and the dad having the B blood group. Now, what are the genes labeling of ABO types? The gene controlling the blood type has been labeled as capital L after the discoverer Landsteiner, or I from isoagglutination. Allelic forms of ABO antigens. The isoagglutinin or I gene exists in three allelic forms: I to the power of A, I to the power of B, and I to the power of O. IA specifies A antigen, IB allele determines B antigen, and I to the power of O allele specifies no antigen. Who are secretors? Individuals who possess these antigens in their fluids, such as the saliva and other body fluids, are called secretors. Non-secretors only has food-sensitive antigens in the blood, so they have no antigens in the saliva, whereas secretors have food-sensitive antigens in the blood, saliva and other body fluids. So these antigens can be detected in the saliva. 80% of the people are considered secretors. Their blood type antigens are found in high concentration in their body fluids, such as saliva, semen, vaginal secretions and gastric juice. If you are a secretor, you will have a higher concentration of A and B antigens than does your blood. With the advent of DNA, the secretor evidence is not as important as it was once considered. Now, what is the enzyme produced in the ABO blood group system? Each allele, I to the power of A and I to the power of B, produces transferase enzyme. I to the power of A allele produces N-acetyl galactose transferase and can add N-acetyl galactose amine. I to the power of B allele encodes for the enzyme galactose transferase that adds galactose to the precursor, that is H substances. In case of I to the power of O allele has no terminal transferase enzyme and it is not produced, hence cannot add N-acetyl galactose or galactose to the precursor. Which blood group allele is dominant? From the phenotypic combinations, it is evident that the alleles I to the power of A and I to the power of B are dominant to I to the power of O, but co-dominant to each other. That means I to the power of A is equal to I to the power of B. So how can we represent their hierarchy? Their dominance hierarchy can be given as I to the power of A is equal to I to the power of B, which is greater than I to the power of O. Now, what are the genotypes of blood group? A child receives one of the three alleles from each parent, giving rise to 6 possible genotypes and 4 possible blood types, otherwise called phenotypes. The genotypes are I to the power of A, I to the power of A, I to the power of A, I to the power of O, I to the power of B, I to the power of B, I to the power of B, I to the power of O, I to the power of A, I to the power of B, I to the power of O, I to the power of O. The phenotypes of the blood groups, 4 phenotype blood groups are A, B, AB and O. Whichever blood group contains A or B or AB are dominant alleles. Now, according to this table, we can see the genotype, the ABO blood group phenotype, the antigens present on the RBC, the antibodies present in the blood plasma. If you take I to the power of A and I to the power of A, the blood group phenotype is A blood group, and the antigens present on the surface of the RBC are A antigens. The antibodies that are present in the blood plasma are anti-B antibodies. Next is I to the power of A, I to the power of O alleles. The blood group present is A, since the dominant allele is A, and the antigens present on the RBCs is A, and the antibodies that are present in the blood plasma is anti-B. Now, I to the power of B and I to the power of B, the blood group present is B blood group, type B, and the antigens that are present on the surface of the RBCs are B, and the antibodies that are present in the blood plasma are anti-A. I to the power of B and I to the power of O, the dominant allele is B, hence the blood group is B, and the antigens present on the RBCs is B, and the antibodies present in the blood plasma is anti-A. In the genotype I to the power of A and I to the power of B, the blood group, both are dominant alleles, hence the blood group is AB, and the antigens present on the surface of the RBCs are A and B. The antibodies that are present on the surface are neither A nor B. So it has no antibodies on the surface. When you come to the genotype I to the power of O, I to the power of O, since there is no dominant allele A or B, the blood group is O. So neither the A nor B antigens are present, whereas the anti-A and anti-B antibodies are present. You can see in this that in blood group A, the surface antigens in the membrane of the RBCs is A antigen, and the antibodies that are present in the plasma is anti-B. In blood group type B, you can see on the left side the surface antigens are B antigens, whereas the antibodies that are formed in the plasma are A antibodies. In blood type AB, the surface antigens of the RBCs present in the membrane are A and B, but there are no antibodies formed in the plasma. In blood group type O, there are no antigens on the surface of the RBCs in the membrane, whereas the antibodies A and B are formed. Let us discuss about the safe transfusions. When you take the phenotype A blood group, the genotype is I to the power of A, I to the power of A, or I to the power of A and small I, or I to the power of O. The antigen on the red blood cell is A. The blood can be given to A blood group and AB blood group, and it can receive transfusion from A blood group and O blood group. If you take the next blood group B, the genotype is I to the power of B, I to the power of B, or I to the power of B and I to the power of O, or small I. The antigen that is present on the surface of the red cell is B. It can give blood to B type and AB type, and it can receive blood from B group and O group. If you take the third type, AB group, the genotype is I to the power of A and I to the power of B. The antigens A and B are present on the surface of the red cells. It can give blood only to AB, but it can receive blood from A group, B group, AB group and O group. Hence, it is called a universal recipient. In the phenotype O, which does not have the A antigen or B antigen, and the genotype is I to the power of O, I to the power of O, or two small i's, then it can give blood to A, B, AB and O blood group. So it is called a universal donor, but it cannot receive blood from other blood groups except O blood group. Let us discuss about the blood group antigens in animals like chimpanzees, gibbons, orangutans. Antigens similar to those found in the human beings have been recognized in the blood of other organisms. Chimpanzees have A type of antigens similar to that of the humans. In gorilla, A and B type of antigens are present. In orangutans and in gibbons, A, B and AB antigens have been found. When you take the chimpanzee, the number of chromosomes is 132. About 88% of the animals are A blood group and 12% of the animals are O blood group. In gorilla, chromosome number is 17. The B group is 88% and the A blood group is around 12%. In orangutan, the number of chromosomes is 22 and the AB blood group is around 23%, the B blood group is 45% and the A blood group is 32%. In gibbons, the number of chromosomes are 14. The AB blood group is around 14%, the B blood group is 72% and A blood group is around 14%. In New World monkeys called platyrrhina and lemurs, they have a substance similar but not identical with B antigens that is found in the humans. In cats, 3 blood groups have been distinguished in cats with a genetic system similar to that of the humans. Now when you take the blood group genes in cats, you have blood group A, they have gene 1 is A and gene 2 is A, so they are homozygous. When you take another blood group A, which is heterozygous, which has a dominant gene A and a recessive gene B. In blood group B, you have the recessive allele B and the another recessive allele B. So it is homozygous. So most cats are either blood group A or B. This is determined by a single pair of genes. The gene for blood group A is denoted by capital letter A is dominant over the gene for blood group B, which is denoted by small letter B. For a cat to be blood group B, it must be homozygous, have 2 B genes. A blood group A cat may be homozygous, 2 A genes, or heterozygous, 1 capital A gene and 1 gene which is small b. Now, something about these felines. AB blood type system is found, and the types that are found are A and B, and AB group is very rare. They are naturally occurring alloantibodies to other blood groups. So there is no universal donor in cats. When you take the blood group type A cats, weak anti-B alloantibodies or weak reaction occurs if transfused with B blood. In type B cats, they have high anti-B alloantibodies. So there is severe reaction if transfused with A blood. Now, what are animal secretors? The secretors or antigens found in the body fluids can also be detected in animals in tears, saliva, urine, semen, gastric juice, and in the milk of animals. So today we discussed in genetics the principles of inheritance and variation part 1 about the multiple alleles regarding the human blood groups and the blood group antigens in animals. Thank you very much. Kindly subscribe, like, share and comment the channel Read Med Prep Academy. Log on to readmedprepacademy.com. Our Facebook ID Read Med Prep Academy. Email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you.